really excited about this study that we just completed, in which we were for the first time able to track single neurons as they're kind of being placed within the brain. The field has known for many years that these neurons are very precisely placed. But how that precise placement happens is not really well understood. We were able to collaborate with physicists to develop these microscopes that enabled very gentle imaging throughout development to be able to capture those early events. And let me show you how these videos look. We can see early on the first neurons appearing and then neurons growing out into what will be the brain. And then we were able to label single cells and understand how is this cell selecting its placement. And then it was through a process that we called retrograde zippering. It's like, it looks very much like zippering backwards, the way that you close a gene, for example. Here, we're seeing this blue neuron, and it's gonna zipper backwards, starting at that point where that green arrow is, as to meet that other pink neuron that is in the correct neighborhood. You can see it actually closing the gap, that black space there, as it's now meeting the correct neighborhood. This is very important. What we're able to do with this microscope is track those events as they're actually occurring and ascribe specific genetic functions to these molecules that we're identifying in that sequence of events. If your car breaks down, the reason that a mechanic can actually fix the car is because there was an engineer that built the car and they understand how the car works. So over here, we have a system that we didn't build. We cannot ask the engineer. So we have to reverse engineer how it works to be able to understand first how it functions and then how it functions incorrectly, which has implications to diseases such as autism, neurodevelopmental disorders, mental retardation, etc. But it's a fundamental question that we need to understand if we have an aspiration to be able to understand how these circuits form and how they form incorrectly in neurodevelopmental diseases.